Hello, I'm Racinda Sherman, Program Manager of Data Use and Research at NACER, the North American Association of Central Cancer Registries. And I'm going to just present a bird's eye view of the role of cancer registries in investigating potential cancer clusters, starting with a brief overview of what cancer surveillance looks like in the US, and then briefly talk about issues cancer registries encounter when supporting investigations into potential cancer clusters and applying the CDC ATSDR guidelines. And then some potential ways that cancer registries can be leveraged to incorporate the genomic and biomarker data that we're interested in today into cancer research. So in the US, we have a national system of cancer surveillance, but it's actually comprised of state-based or regional cancer registries. But the primary role of cancer surveillance is to collect cancer incidence data on all persons that are diagnosed with and treated for cancer in the US. And the data is collected so that we can track the burden of this disease over time, so that we can plan for treatment for the population, as well as to guide cancer control and prevention activities. And cancer surveillance data is then also able to be used to support cancer research. And so all of those together have the overarching goal to reduce the burden of cancer in our communities. So I am representing NACER, a nonprofit, the North American Association of Central Cancer Registries. And we're a member organization that supports all central cancer registries in the US and also in Canada. Specifically, NACER develops national data collection standards for cancer registries. We also certify cancer registry data for quality, and we support the use of cancer registry data in a scientifically sound and for the purposes of public health research. And the cancer registries support a variety of data uses and research at both the local and the national level. Cancer registries, they generate all of the annual cancer rates and trends that are reported. We provide data for cancer control and research, as I had mentioned. We do respond to community concerns about potential cancer clusters, and that's often done by calculating observed versus expected rates. But we also standardly produce other rates by geography. At county and sometimes at sub-county. And over the last decade or so, we've been able to produce a lot more data visualizations of the cancer data on maps. And the role of NACER really is to work with these members in a committee structure to address data collection and analysis issues. To be able to support the work of the cancer registry. So we support data use and research through, again, this committee structure. Various work groups have members from the cancer registries, and they just have specific aspects of data collection and analysis that they work on. So for instance, we do have a number of NACER work groups that actually focus on improving the use of cancer registry data for geospatial analysis, which of course is what is needed for cancer cluster investigations. So the data cancer registries that collect, of course, underpin cancer cluster investigation. But as everyone that's been involved in a potential cancer cluster investigation knows, there are a number of challenges that make these investigations difficult. What we really want to aim for is this example here on the right. The image here really represents a success story. One of the first publications of national cancer maps showed unusually high rates of lung cancer diagnosed along coastal areas. Those are the red pink areas that you can see on the map. This prompted further epidemiologic study, and this is how the relationship between asbestos and mesothelioma, which is the specific type of lung cancer associated with asbestos exposure, how that relationship was identified in the shipbuilding industry. But unfortunately, most investigations are not as successful in identifying causal exposures or even identifying clear guidance for a cancer control intervention. There are a number of methodologic challenges that can result in studies that both fail to establish causal relationships and while also simultaneously failing to rule out potential environmental exposures. Sometimes an elevated rate or a higher burden of cancer may be found in a community, but it's actually unclear if it's meaningful. And in this case, I mean meaningful, meaning that it's caused by a shared exposure that we could actually mitigate. And sometimes we can't distinguish those from clustering that happens merely by statistical chance. And perhaps there's a common exposure, but what that exposure is might actually never be established. And so these can be incredibly frustrating for all involved. Sometimes, unfortunately, the identification of a higher burden of cancer with no clear next step for cancer control could actually have unintended consequences. So for anyone who followed the pediatric cancer cluster investigation of the Acreage community in Florida, you know that a higher rate of pediatric brain cancer was identified, but then there was no specific environmental exposure identified. So there was no clear actionable next step that could reduce the burden in that community. And then unfortunately, the community not only didn't get answers on the cause of cancers in their children, but then they suffered financial losses as the value of their homes plummeted due to the perception that the community was unsafe, causing additional burden to this community that's already suffering. And while these investigations are important to advance our understanding of cancer risk and exposure, sometimes there's already a clear exposure to a known carcinogen or other health hazard. But the burden of proof is generally placed directly on the community, which delays proper response to the hazard. There's no reason that we need to wait for a long-running cancer cluster investigation because waiting for the results of the study is potentially causing further harm and also lack of accountability for the mitigating the exposures. So at the start of today, Stephanie Foster has provided a nice overview of the CDC ATSDR guidelines and their recent updates. And these guidelines are a really critical resource to try to handle some of the issues that registries and health departments encounter when they are trying to respond to a cluster investigation. And there are two components in the new guidelines that registries really support as a potential best practice, but they may not actually be able to incorporate them into their registry operations at this time. So NACER has a long history of supporting cluster detection techniques and using these methodologies to proactively advance our understanding of the local patterns of cancer. But we also understand that there's no one best method to apply. And using different methods can actually often result in conflicting results. So that makes it challenging to understand when you are performing cluster detection and other proactive methodologies. It's hard to understand how to interpret what the results may be. And then additionally, while some registries and health departments may actually be able to incorporate the annual review of unusual patterns, it is important to note that both this proactive cluster detection and the continuous monitoring part are highly resource-intensive activities. And right now, the reality is that registries are not funded for these activities, and they generally already operate with very lean financial resources. So while we, the NACER members, the cancer registries, really do have expertise and interest in this area, the ability to adapt the new recommendations will likely be dependent on obtaining additional funding. So, but back to the specific topic of today, one of the methodological limitations for potential cancer cluster investigations is lack of data, all sorts of data, but particularly exposure data, which would include the genomic and biomarker information, which of course can potentially reveal genetic risks as well as indicate potential environmental exposures. But the vast majority of that data in the cancer registry, well, the vast majority of the data of the cancer registry comes from clinical records where genomic information is not routinely collected. Registries do collect some limited biomarker data, but these data are only collected after the clinical importance of that marker has been established. So there's a bit of a delay. It's not before that information would be incorporated into the cancer data. But we do have a NACER work group, and that work group is tasked with assessing the feasibility of collecting genomic data. That work is just beginning, but there is a wider group outside of NACER Genomics, which is working to standardize the collection of the genomic data so it can be standardly incorporated into clinical and other health data. So one of the uses would be to include the data in cancer registries. So one of the looming issues, of course, is that these types of test results are still reported as a PDF, making it difficult to abstract the critical information and to use it outside of individual clinical care. And so the genomics group has developed an HLL standard to make the data collected standardly and able to be transferable electronically into systems like the cancer registry. Great. And then another way to include genomic and biomarker information with the cancer data is through a NACER system called the VPR, or Virtual Pooled Cancer Registry. Virtual Pooled Cancer Registry Cancer Linkage System is its full name. And this is a service that assists researchers to link their cohort data to cancer registry data. The VPR system can support and has supported traditional occupational cluster studies on a national level. The strength of this is that it provides a standard, scientifically standard way of linking the data so that you can get consistent results, but also coordinates the data on a national level rather than on a state-by-state basis. This approach is study-based, and we do have the infrastructure already established. And this could be a more efficient way to incorporate the genomic data for cancer research rather than collecting and storing it in the registry data that would be a much slower process. However, the VPR is also currently limited in capacity as the linkages also are resource intensive, but the structure is in place that we would be able to scale it up if there was appropriate funding. And that's really all I have. We had a really short time, but I will be available for any questions after the other panelists' presentations. And you can always connect with me after the workshop at this email here. Thank you.